5-carbon compound, you add carbon dioxide to it, you make a 6-carbon intermediate, which then splits to make a 3-carbon compound. So you start with 3 5-carbon compounds, you add 3 carbon dioxides, total of 18 carbons, you make 6 3-carbon compounds by the end of carboxylation, 18 carbons, a gain of 3 carbons. The next step, reduction. You're going to use ATP and NADPH, and you're starting with this organic acid, and you're going to turn it into an aldehyde. That's the whole point of reduction, is to turn it into a useful aldehyde. That aldehyde is a 3-carbon sugar that can be the basis for making everything else the plant needs, like glucose, fructose, maltose, sucrose, all of those other sugars, including starch and cellulose. So that's the point where some carbon will be exported out of the cycle. So this diagram is showing you how the reduction step is working. So 3-phosphoglycerate, you have an energy investment phase here. ATP is used, you add a second phosphate group there. And then when you add the electron from NADPH is oxidized to NADP+, and then the phosphate comes off, and you end up with the aldehyde instead of the what used to be a carboxyl group here. So that carboxyl group, through these 2 steps, is turned into an aldehyde group. Now the aldehyde is the sugar that is the product of photosynthesis. So all those books that tell you, you use carbon dioxide and water with the energy from light and you make glucose, no. You do not make glucose, you make glyceraldehyde 3-phosphate. Yes, sir?
The phosphate group comes off from 5-phosphoglycerate when the electron is added?
So the reduction step does remove the phosphate that was added. So you have to energize the molecule before the reduction reaction actually goes forward. So both steps are required for reduction. So far, so good? All right. The regeneration step is the most complicated part of the cycle. So some of the glyceraldehyde 3-phosphate is exported, and then some of it is going to be used to regenerate the RUBP. Because if you don't have RUBP, the whole cycle stops. You can't add any more carbon dioxide to the body of the plant. Now this includes 4-carbon compounds, 5-carbon compounds, 6-carbon compounds, and 7-carbon compounds. It's energetically expensive, it requires ATP, so more ATP from the light reactions is going to be used in the Calvin cycle to regenerate RUBP. And these are the steps, and I do not expect you to at all know the details of this diagram at all. But I do want you to follow it with me. So you're starting with 6 3-carbon compounds at the top. So everybody just look up. Everyone just look up. Start with 6 3-carbon compounds at the top. One of them is going to be exported. So 3 carbon dioxides came in, 1 3-carbon compound is harvested. That's the gain of the 3 carbons for the plant. The other 5 3-carbon compounds are equivalent to the 3 5-carbon compounds you started with. But now you want to regenerate those 3 5-carbon compounds. So that's what all of this is about. Take a 3-carbon compound and another 3-carbon compound, you have a 6-carbon compound. Add another 3-carbon compound, you have 9 carbons. That forms a 5-carbon compound and a 4-carbon compound. The 5-carbon compound could be turned into RUBP. The 4-carbon compound is still useless. Take a 4-carbon compound and another 3-carbon compound. Makes a 7-carbon sugar. Take the 7-carbon sugar, add another 3 carbons, you make 2 5-carbon compounds. Those 2 5-carbon compounds can now be turned into RUBP. So you end up with 1, 2, 3 RUBPs. Now these steps do require ATP, so notice the use of ATP down here to make RUBP. And that's as much as you need to know, unless if you take an upper level plant physiology class, which is not saying much.
So how many 3 carbons do you need for RUBP?
RUBP has how many carbons? 5. So you had 3 5-carbon compounds, and now you're starting with 5 3-carbon compounds, same number of carbon, 15. You're just rearranging those 15 carbons to regenerate the 3 5-carbon compounds. 1 3-carbon compound was exported, and that's equivalent to the 3 carbon dioxides that came in. Bring in 3 carbons, export 3 carbons. The other 15 carbons just keep getting recycled. Any other questions? Great. So the net effect, 3 carbon dioxides come in, 3 turns of the cycle, you export the equivalent of 1 3-carbon compound, you maintain your original acceptor molecule, the RUBP, you require NADPH and ATP molecules, and the efficiency of the cycle is actually quite high, 88%. Efficiency of photosynthesis as a whole, not as high, and we're going to see why. There's a problem with oxygen. Photosynthesis makes oxygen, but oxygen is also a problem for photosynthesis. And that problem becomes much worse if the leaves have to close their stomata. So if they're drying out because it's warm and sunny and dry, lots of light reactions happening, but they can't afford to keep their stomata open because they're going to dry out, lose lots of water, then oxygen becomes a bigger problem. So one thing you should know is that this enzyme, Rubisco, the most abundant protein on the planet, happens to have binding sites at its active sites for carbon dioxide, which also happen to bind oxygen. So oxygen can bind to the active site of Rubisco. This is a problem. Now the good thing is that oxygen doesn't bind that efficiently. So the enzyme is approximately 80 times more efficient at binding CO2 than it is at binding O2, because it's really designed for binding CO2. But CO2 and O2 kind of look alike. And one is bigger. Hopefully you know which one is bigger. They're both hydrophobic, perfectly symmetrical, and even though this one, the top one, CO2, binds to the active site much better, that one kind of fits too. Not as efficiently, but it fits. And if you look at the atmosphere, how much oxygen do we have in the atmosphere? Approximately 21%. And how much CO2 do we have right now? Approximately 400 parts per million. So this is parts per 100, that's parts per million. So a lot less CO2 in the atmosphere than O2. Now the atmosphere isn't everything because the CO2 and the O2 have to go into solution for Rubisco to actually interact with them, but because there's so much more oxygen in the atmosphere, you will end up with more oxygen in the solution in the stroma because there's a lot more of it. So it's a good thing that the enzyme does not have high affinity for oxygen. Because this is what happens when the enzyme actually reacts with oxygen instead of CO2. So you still have your 5-carbon RUBP, but instead of adding a carbon by adding a CO2, you're actually attaching an oxygen to it. And that splits the 5-carbon compound into a 3-carbon compound and a 2-carbon compound. So you're not getting 2 3-carbon acids out of this, you're getting 1 3-carbon acid and a 2-carbon useless compound. So the phosphoglycolate is a useless 2-carbon compound that the plant can't really use. So when oxygen reacts, not only are you not gaining any carbon, you're potentially losing 2 carbons you already had. So it's a big waste. This is the oxidation reaction by Rubisco. Remember, Rubisco is ribulose bisphosphate carboxylase oxygenase. This is the oxygenation reaction, which is the total waste of carbon for the plant. The plant is not gaining anything and it's losing 2 carbons it already had. Now, plants have evolved mechanisms to deal with this. So, before we talk about that, you have to consider that under certain conditions, this problem becomes a bigger problem for plants. So for most plants, which are called C3 plants, because the first product of the Calvin cycle is a 3-carbon compound, the ratio of carboxylation to oxygenation is 3:1 under normal conditions. So even though the enzyme has 80 times more affinity for CO2, because there's so much more oxygen in the atmosphere and therefore so much more oxygen in the solution in the stroma, this comes to 3:1. So for every 3 times that carbon dioxide binds, 1 time oxygen will bind. So 25% of the time, the plants are losing carbon they already had, which is a problem. That's why the efficiency of photosynthesis is not as high as you would think it would be, because oxygen is a problem. But it gets worse when it's warm and dry, because the plants have to close their stomata. And what happens to solubility of gases in solution when temperature goes up? What happens to your soda when you have it warm as opposed to cold? It tastes flat because the carbon dioxide, that is the reason your soda is fizzy, comes out of solution. So your soda is not fizzy anymore when it's warm. The problem is that the solubility of CO2 in solution goes down faster with increasing temperature than that of O2. So as temperature goes up, both CO2 and O2 come out of solution. But you lose more CO2 out of solution than you do O2 for any increase in temperature. So proportionally, you end up with more and more O2 as the temperature goes up, because CO2 comes out of solution faster. That's one problem. The other problem is that plants will close their stomata if they're drying out, which means the Calvin cycle goes on, uses up all of your CO2, and then you have no CO2 left. What is left? Plenty of oxygen in those air spaces in the leaf, 21% oxygen in the air. If you don't have any CO2 left, oxygen will bind. So if it's warm and dry, this ratio keeps going down. So that's a problem. So evolutionarily, plants have evolved ways to deal with this. One way is photorespiration. So plants will use the O2 in this oxygenation reaction, which is a total waste, and then in the salvage operation, they will actually make CO2. And we're going to take a look at that. So the salvage operation is called the PCO cycle. And the PCO cycle involves 3 different organelles. It includes the mitochondria, the chloroplast, and the peroxisomes. And you do not need to memorize this figure, but you do need to pay attention to where the red arrows are. So we start at the top here. Here's RUBP. And instead of reacting with carbon dioxide, it's reacting with oxygen, the oxygenation reaction. And to balance everything, we're multiplying everything by 2. So 2 RUBP compounds, 10 carbons total, 2 oxygen molecules. That gives you 2 phosphoglycolate. That's the 2-carbon compound. So that's 4 carbons. And 2 PGAs. That's the 3-carbon compound. So 6 carbons here, 4 carbons there. You start with 10 carbons, you still have 10 carbons. The PGA is the 3-carbon acid. That's useful. That can be used as is. The phosphoglycolate, totally useless. So if they don't do anything to it, it's a loss of carbon. Now, if you follow the reactions here, notice down here you're losing carbon dioxide. So you have 4 carbons from the 2 phosphoglycolates going through the cycle. You lose 1 carbon. Start with 4 carbons with the 2 phosphoglycolates, you lose 1, you're down to 3 carbons. And the 3 carbons are turned into PGA, which is the useful form. So instead of losing 4 carbons, the plant loses only 1. So that's not so bad. So you're salvaging 75% of the potential loss. You're only losing 25% of the potential loss. That's not so bad. but it does cost ATP.